Hello, everyone. Thank you for coming to this week's video lecture. Today, we're going to be talking about bacterial diseases. So in the past, we've talked about generally how bacteria invade the body, how they interact with the body, and then we've talked about the immune response and how the body interacts with the pathogens, but very generally. And now we're going to do an overview of different diseases in a more specific way. And first, we're going to talk about staphylococcal and streptococcal diseases, pneumococcal and neiserial diseases. So with staphylococcal species, these are, again, those round cockal bacteria that are typically gram-positive, and they're in these grape-like clusters or these staph-like clusters. And they're considered, there's a couple of different species that are medically relevant. This is like Staph aureus epidermidis and like Saprophyticus, I think. They grow commensally on the human body and typically have a distinct coloration and can tolerate high salt. And so usually you can grow them on mannitol salt agar, which is a high concentration of salt. So you can kind of identify these staphylococcal species because of that ability to withstand high salt. Additionally, Staphylococcus aureus is one of those main species that you hear about that cause disease. And this is because there are virulence factors that it has, like coagulase, that when it gets into your blood, it can cause the plasma, the fibrinogen to convert to fibrin, and it creates these clots within the plasma of the blood, creates these fibrous networks that the bacteria can live inside and grow in. And it can make it harder for the immune system to the immune cells to actually get into that mesh network. And it kind of hides the infection from the body. We also have exfoliative enzymes so that if you have a skin infection of Staph aureus, it can secrete this exfoliative toxin onto the skin, and it can break down the junctions between your cells, and it can cause the bacteria to get deeper into your cells, into your skin, penetrating that wound further into that tissue. Staphylococcus aureus and other staph species are found commensally on the skin. They're a typical part of the human microbiome and in the nose. And they can also just be transferred onto the environment because of us interacting with the environment. And so there are places where these bacteria are commonplace just because we are commonplace, like in hospitals, where they can actually uptake genes that give them antibiotic resistance. One of the most notable one is methicillin resistance, Staphylococcus aureus, or MRSA, which is a really strong antibiotic resistant form of Staph aureus, and it is a major concern in nosocomial infections where you get an infection from a hospital setting. So staph diseases typically occur on the skin because they're found on the skin. And usually when it's either there's an overgrowth in it, and the immune system that is in that area can't fight off of that overgrowth, and it creates these infections. Staph gets into like a hair follicle, and it creates folliculitis. And if that folliculitis doesn't resolve, it can be a furuncle. And if you have a lot of furuncles, they can be a carbuncle. And so furuncles are these infected like hair follicles that seem to be kind of a more chronic kind of situation. And when you have a lot of them together, they create these large masses of inflammation called a carbuncle. You can also have impetigo, where the staphylococcus is on the skin and is causing the sloughing off of that skin. Remember that exfoliative enzyme causes the breakdown of the junctions of the epithelium. It causes sloughing off, and it creates this kind of scarry-like appearance. And it also creates inflammation. And so depending on if this is done locally or systemically, depends on what kind of interactions and symptoms you're going to be getting. Again, you can also have even more aggressive infections with staph species, like in your bones, in your blood, and in your lungs, where you have osteomyelitis, where you have in your growth plates of your long bones, the staph can become colonized. You can have bacteria infecting other parts of your body, or septicemia, where you have it in your blood. You can have pneumonia, where it grows inside of your respiratory tract. You can have, because there are these toxins that are being produced by Staph aureus, you don't always have to consume the bacteria itself. You can also be intoxicated by the toxin. You can consume just the toxin, not the bacteria, and you can still get effects from that. This can be like just some food poisoning. It can be from enterotoxins. And it can cause some gastrointestinal distress. So you might get some vomiting, you might get some pain, you might get diarrhea, depending on where it is. Usually your symptoms are going to be kind of, where is that toxin being released? So if you have your toxin, if you're consuming the toxin, your toxin's first being exposed to like your stomach and like the upper GI tract. And so like you're going to have like pain and gas and like vomiting. But if you like consume the bacteria, and the bacteria goes all the way through your digestive tract and then excretes the toxin like midway through your gut or at the back end of your gut, you're gonna be getting diarrhea and you're gonna get pain and gas and things like that. But you might not get vomiting because it's already past the like stomach and things like that. And so you have to kind of think about what you're consuming and how it's going through your body to kind of elucidate or kind of think about what these symptoms could be. And so anyway. So you have the food intoxication, where you can like consume the bacteria or the toxin, and it can cause you to have gastrointestinal distress or food poisoning. You can also have toxins that are being released that affect the skin. We talked about how impetigo kind of sloughs off the skin a little bit. Staphylococcus scalded skin syndrome is another similar form of this, where it looks like you have these large patches of inflammation and then some sloughing off of the skin layers, where it looks like you've just had this really severe burn. And you can also get toxic shock syndrome, where you have systemic inflammation and systemic shock. So then we have the consuming of the enzymes that the staphylococcus creates, and that's intoxication. And then we can talk about how we can identify staphylococcus species. And we've done this kind of in lab, where we are able to put peroxide onto the bacteria itself. And if that bacteria has the catalase enzyme, it's able to decompose that hydrogen peroxide and create water and gas. And this creates bubbles. So you can see that this species here is catalase positive. It has the enzyme that can decompose that hydrogen peroxide. And so we can utilize something like that. We can utilize the coagulase test, where we put the bacteria in some plasma to see if it creates that fibrin clump. And we can also test the different medias that it grows on to specifically identify what bacteria it's growing. And we can also visualize it under the microscope. And so for the most part, these bacteria are commensal on the body, but there are times where they are more problematic for like immunocompromised individuals or for infants. And then also sometimes they can have antibiotic resistance. And so that's going to complicate treatment when they they essentially are gaining virulence factors. And remember, virulence factors aid in the pathogenicity of the bacteria. And so if you gain virulence factors, you're probably gaining pathogenicity, and you're going to be able to create an infection a little bit easier. And so MRSA is a bit more of a risk and harder to treat because of that. And so that's where we're going to have to like be on the lookout for those kinds of situations. We treat it with cleaning and with antibiotics, and making sure that you have good hygiene, and making sure that you're not like leaving spots on your body to just like overgrow, essentially. So like wash your hands and like wash your body and wash your wounds, like within reason. I'm not a doctor, so like listen to them first, but I guess I'm a doctor, not a medical doctor. So streptococcal species is a different kind of is a different group of bacteria. These are round bacteria that grow in chains instead of clusters. Remember, staph are those grape-like clusters, and strep are those long chains. And these streptococcal species are typically commensal in the nasal passages and on the throat, sometimes on the skin, but most of the time it's in the nasal passages and in the throat. So for these streptococcal species, there are a couple of different categories of these streps. You have A, B, C, and D groups. The infection that you can get is different depending on the group that you're infected with. So when we look at streptococcal species, we usually sample from the throat and put it on blood agar, and we look at how can it lyse the blood cells that are in that agar plate? Can it completely lyse them with beta hemolysis, or can they only partially lyse it with alpha hemolysis? And if it's beta hemolytic, we go through some tests to make sure and see if it's group A strep or group B strep. And if it's group A strep, this is going to be Streptococcus pyogenes, and this is what you're going to typically see when you have strep throat as like a sickness. Where strep throat can have different virulence factors and can cause some like downstream effects like rheumatic fever and scarlet fever and things like that. Alpha hemolytic bacteria, that is streptococcus, can be like Streptococcus pneumoniae and group D viridans. So the beta hemolytic species like Staphylococcus pyogenes are going to be what you find inside of the back of your throat and in your nasal passages. These are going to lyse your blood cells with some of its virulence factors, its hemolysin, and it's also going to create tissue damage as it creates a more favorable environment, and it collects like a buildup of white blood cells and things. That's where you like those white pustule things on the back of your throat are coming from, is the growth of bacteria, the accumulation of tissue damage, and the white blood cells. And for the most part, this strep pyogenes is going to be like, for the most part, a parasite. It's not going to be considered commensal. However, there are people who have it as like a carrier. They might be asymptomatic, but for the most part, most people, when they have this, they're going to have like an overgrowth of this streptococcus pyogenes. They're going to have an infection of some sort. However, it still is still found in the throat of people, and that's how it kind of gets around. And then contacts through just like lip smacking with direct contact, droplets through breathing, sneezing, talking, and then through preparing food and through like extra to contact. Maybe I put my pin in my mouth and for some reason put it down for someone else to grab, and then someone grabbed it. That would be really gross. There's probably better ways, like you put your hand near your mouth and then you put your hand somewhere, or you shake their hand or something like that, right? Like somehow it has to get from your throat to their throat, and there's lots of different routes of transmission. And usually how it gets into the body is usually through the nasal passage or through the mouth. Occasionally it can infect the skin. But and so in that case, it's just going to be entering into like a wound or a break or something like that. But for the most part, it's going to be in the throat. That's what you're going to like think of the most. And then it creates that damage within the tissue in your throat. Those are those white pustules that I was talking about. That's those tissue damage. And then you can have impetigo where it's growing on the skin. You see that more on infants. And for the most part, you're going to treat this with like antibiotics. In some cases, it can infect the skin. It can, because of those virulence factors, it can cause necrotizing fasciitis, where you're starting to literally break down the tissue that's within the body that it's infected in. I'm going to change the slide real quick just so that we're not like looking at that anymore. Something that's like really interesting about this necrotizing fasciitis is that you're degrading the tissue, and the tissue is starting to break down, and then it loses its oxygen. You are killing the blood vessels that are coming in and around it, so you can't deliver oxygen to it, and then it's just like a mass of dead cells. And so like oxygen can like permeate through diffusion on the surface, but it really needed to get oxygen inside, blood inside of it to get that oxygen inside of it. But because it's all dead, it's not doing that. So the environment within that tissue is actually anaerobic without oxygen. And so this is a favorable environment for bacteria that would typically never get a chance to grow on the human body, except for within the gut, where it is anaerobic, right? Most of the time, your skin and in your nasal passages, all of those places, you're not going to have oxygen deprived environments. So you're going to not have bacteria like Closidium perforans growing inside of that sore. And that, for instance, C. perforans is what causes like gangrene. But that only is because C. perforans is an anaerobic bacteria, and it can only grow in that site because of becoming anaerobic because of the bacteria that's growing on it. Strep can also cause long-term effects like scarlet fever and rheumatic fever and pneumonias if it can travel into the lungs. But it can also cause like heart damage and kidney damage depending on where the bacteria is growing. And then also there are some studies talking about that the surface sugars that are on top of the streptococcal pyrogenes bacteria look similar to our own proteins and sugars that we have, and we can create antibodies that react and hurt ourselves. Those would be autoreactive antibodies, and they can cause damage. And those things are, when you see something like it causes chorea, that's what it's meaning. That's what it's talking about typically. This right here, this chorea. Anyway, and so like that's what are like movement disorders. So then you treat this again with antibiotics. You want to do it quickly so that it doesn't progress to this like rheumatic fever and scarlet fever stages, because those things can lead to chronic complications later down the way in life, like heart problems. The Viridians group is a set of bacteria that like it's diverse, and it can cause like tooth decay, gum disease, and genital tract infections. And so S. mutans is what we are typically associate with the formation of the plaques within our skin. Nope, our plaques within our teeth that allow for that fermentation to occur that creates those cavities. So these viridians can also get inside of the circulatory system and cause issues elsewhere in the body. So for instance, if you have some of these in your mouth and you brush too hard, or you have an abscess, or you floss and it gets into your gums, into your circulatory system, it can go elsewhere in your body and create similar infections like a tooth abscess, just somewhere else. And so somewhere else could be your heart, and that would be subacute endocarditis, where we're causing these little growths to happen on the heart, which causes damage. So these pneumococcal species are these diplococcus arrangement, or these short chains. So if you remember, the strep had really long chains, where these pneumococcals are really short chains. And they grow in the nasal passages and the throat. They require a really enriched environment with different substances, and those are found on the body. And so they are what are known as fastigious, where they're hard to grow outside of the body and require special media. And And because they're in the nasal passages and in the throat, they can cause infections in the lungs, they can cause infections in the nasal passages. And then also, if you remember that estrogen, that what's that tube called, that I can see the word, but it's that etrian tube or whatever that connects your mouth to your ear, that can also be infected. And so this streptococcal pneumoniae can actually infect that tube and infect the inner ear and cause inner ear infections, that otis media. And it can also travel into the meninges, the membranes that surround the brain and spinal cord, and infect those as well. And so this Staphylococcus pneumoniae can cause meningitis, it can cause ear infections, it can cause pneumonia, and it is found across people. And it's a typical bacteria is a common bacteria that you find in people. And it doesn't live well outside of its environment. It can get inside and infect the inner ear, or it can go into the lungs and infect the alveoli. You can usually diagnose this by culturing the sputum on special media, or you can do a gram stain to visualize these diplococcus and short chain. You can treat this by antibiotics or through preventative measures like the conjugated vaccine. I will say that it's really interesting that this conjugated vaccine shows to be pretty effective, but there's just like a small subset of people that can't form a response to this conjugated vaccine that just doesn't seem to be working for them. And so people are still trying to figure out why, but it seems what that's called is called specific antibody deficiency. So you are lacking the antibodies for this pneumococcal surface proteins. So then finally there's neiserial diseases, and this is like neiserial gonorrhea or meningitis that causes infections in sexually transmitted diseases, or it can cause meningitis. So Neisseria species are fairly uniquely shaped. They have this like bean-shaped diplococcus bacteria. And the two main ones that you're going to hear about in the clinic would be gonorrhea and meningitis. Meningitis, gonorrhea, and meningitis. And so gonorrhea is one of the most top infections of STDs, and it has these virulence factors like fimbriae that helps with attachment to prevent sloughing off of the bacteria. And it can prevent cells from engulfing them. And also because they live inside of the mucous membrane of these of the tissue, IgA, if you remember, IgA is secreted into the mucus layers between the outside of the epithelium. And so the Neisseria gonorrhoeae has virulence factors that will chop up and make the IgA inactive. And so it essentially provides protection within this mucus against this IgA that would typically be invading that mucus to try to clean it up. Typically, you only need 100 to 1,000 of these cells to become infectious, and it doesn't survive very long outside of the body. It can be asymptomatic, and it creates this discharge and inflammation. It can cause other infections because it can compromise the mucosal immunity, and it can cause pregnancy issues as well. You can also get infections outside of the genital region, like the anus and the pharynx and in the eyes. You can also get it in the blood and in the joints, but for the most part, that's going to be like in rare cases where you have severe hemorrhaging and infection at the same time, which does happen. We also have gonorrhea in children. So you give birth, and if you are infected with gonorrhea, that gonorrhea can get on from the vaginal canal onto the infant. I always call them fetuses, even though they're fully formed babies. And so the gonorrhea could get onto the baby through the birthing process, and it can infect their skin. And if it gets into their eye, it can cause blindness. This can be prevented through antibiotic treatment before pregnancy. You could also probably go through like a C-section to avoid that completely. through the birth canal. And then the other thing is, for the most part, young children aren't going to be exposed to gonorrhea. And if they are exposed to gonorrhea, there is probably signs of sexual abuse and it needs to be reported. And so gonorrhea is one of those diseases that is monitored very heavily by medical professionals and government agencies. And so it's a mandated reporting. It's like a disease that you have to report. And so that makes sure that we can keep track of what this disease looks like and who it's infecting and why. And that also helps raise flags when there is suspicion of abuse. We also have, to identify it, you can see the kidney-shaped, these are white blood cells, but if you zoom in, there are the bacteria in those little pink dots. If you zoomed in even more, you'd see that kidney-shaped diplococcal look. And they also can get inside of your neutrophils. So these are your neutrophils. You can tell by the like multi-lobed nucleus. And that also makes it where it's harder for them to die because they can hide inside of the cell. Then we have Neisseria meningitidis, which also has similar virulence factors, but instead it infects from the nasal passages, the meninges around the brain and spinal cord to cause inflammation and damage that way. You can also get that same disease, gonorrhea meningitidis, instead of in the meninges, you can also get it elsewhere in your body. And so if it goes from your nasal passages and it goes to your brain and it can break down the blood-brain barrier, it creates meningitis. Otherwise, it creates this meningococcal anemia, which would be meningococcus, which would be the bacteria in the blood. The emia would be in the blood. And so it's this bacteria getting into the blood and then going into the circulation and causing the same kinds of tissue damages that you'd see in the brain, but outside on the skin. We then have Closidium botulinum, Closidium tetani, and Mycobacterium tuberculosis. So Closidium tetanus, Closidium botulinum, all of these are found within the soil. Typically, tetani is usually you're exposed to it through your environment. Through Closidium botulinum, you're exposed to it through your food. You can classify these diseases as, are they going to be infections that you get just like on your day-to-day life, or are they going to be something that you get from food specifically? And Clostridium species typically have pretty powerful toxins that are used as these virulence factors to progress its disease or its infection. So first we'll talk about Clostridium tetani. So Clostridium tetani is found in soil. It's eaten by animals and is secreted out through excrement. And that's how it goes from one place of the soil to another place in the soil, right? Sometimes it can get onto our food or just like on like everyday objects. And then those everyday objects somehow get into us, whether like a scrape on your scrape with something that was exposed and it had these spores on it, you know, it somehow gets into your body, right? Through that portal of entry. And then it can grow under very specific circumstances, and it can secrete this toxin that can cause systemic neuromuscular issues where you're having severe contraction and tightening of your muscles. And this is typically heard of at first within your jaw and known as lockjaw. And if the infection continues, it's going to become a systemic infection, and it'll be in a whole body contraction of your muscles. And so the special requirements that are required is this anaerobic without oxygen environment. Remember, we talked about how necrotizing fasciitis can be like a first stage of creating an environment that's anaerobic. Well, Clostridium tetani would love that environment, right? There are other ways we can get anaerobic environments than necrotizing fasciitis. Some of this can be through the scars that we get from our body through major scars. It can be through crushed body parts, frostbite, and we just need to make sure that there's no oxygen, and that will allow for these spores to germinate and the bacteria to grow back into its vegetative form, release these toxin, and it can start these symptoms of muscle contraction, lockjaw, muscle contraction, and like these overarching symptoms of the back. But you can tell that this is incredibly painful, and typically because of the amount of contraction and the inability to release and then recontract, you're going to get respiratory issues and cardiac issues. For botulism, we have botulism is again found in the soil and infects typically the food that we find. And if you kind of think about, it's usually low acid food, but because it's found in the soil, when we harvest our vegetables, if we harvest potatoes, things like that, those things are all in the soil. Broccoli, those are near the soil, those things are getting picked, and botulism spores can get into those food items and then get packaged and canned. And if they're not completely sterilized properly, they're being put into this anaerobic environment in the can of low acid and without oxygen. And this is where these spores germinate, create their toxin. And then if we consume that toxin, we're getting what is known as intoxication. There also could be botulism, like botulinum, sorry, the toxin is called botulinum. There could also be the actual bacteria, Closidium botulinum, and we could consume that. But we might kill it through our digestive tract. But if it gets into our lower digestive tract, it'll be able to have that anaerobic environment, and it can create, again, those toxins that are going to give us, instead of contraction, it's actually going to be the inability to contract our muscles. And so this toxin called botulin prevents the release of these acetylcholine vesicles and blocks us from telling our muscles, okay, contract. And this causes us to essentially lose, essentially be kind of, flaccid, to be floppy. And so this causes flaccid paralysis in infants. This is called floppy baby syndrome. And this is where we're not able to contract those muscles, and so we're not able to keep tense, right? And this is particularly a problem within infants where they don't have a completely mature immune system, where they can't fight off those spores completely. And so places that you would find this very commonly would be those low acid foods of contaminated food items, which would be like something that we would get contaminated with. But also things that naturally have these spores like honey, because honey is just pollen that's been collected from the environment. That honey could have botulism spores in it. And the high sugar content prevents them from germinating, and then our stomach acid can kill it. And we can prevent it from going any further if it doesn't get killed with our immune system. But infants don't have all of those barriers completely fully formed yet. And so eating honey can be a potential source of botulism spores and botulism. And we also had like contaminated food for like infants just recently this past year or so. And so it's definitely a bacteria that is a problem in food safety. And so we have to test our foods and make sure that they're fully decontaminated while packaging, making sure that they're sterile in that capacity, but also making sure that it's fully cooked. And then also sometimes these toxins are, I don't think it's in this case, but in some cases, these toxins are heat stable. And so even cooking them isn't going to stop it from causing illness. And so the sanitation throughout the entire process is incredibly paramount for all kinds of food-borne illnesses. We've been able to leverage this botulin toxin as Botox to inject and kind of release the muscles within certain regions, like we have with facelifts and things like that. But it wears off over time as that botulinum toxin degrades and as those tissues recover. So then we have Mycobacterium tuberculosis, which is an acid-fast bacteria. Remember, acid-fast is a specific type of stain that looks for this mycolic acid within the membrane of the bacteria. And typically these mycobacteria are going to be kind of problematic with gram staining. And so even though that their cell structure can be identified as gram positive, when you stain them, they might not be as easy to stain as gram positive, but acid fast would be a good relative way to show that. They're strictly anaerobe, so they have to have oxygen, and without oxygen, they would die. And so typically where we're going to find this is in the respiratory system. And they cause a incredibly horrid disease called tuberculosis. That's just like the silent killer. If you grow tuberculosis, Mycobacterium tuberculosis on a plate, it has these really distinct colony morphologies. And when you look at it under a microscope, it has these very long thread-like chains. Remember, these would essentially be like a strepto arrangement, but they're in such a very long, thin-like characteristic, they are typically considered, they kind of are classified as their like own special thing, but that's just because they look so distinct, right? And so then they have waxy, that mycolic acid as a virulence factor, which helps prevent the absorption and the detection by the immune system and like prevents that phagocytosis. And then once they are phagocytosed, they're not going to be killed by the lysosome. And so you're going to actually get intracellular bacteria. These bacteria just grow inside of the macrophage. It's a very slowly developing disease, but it can also resist, because of that waxy coating, it prevents it from drying out, and so it can survive outside of the body for a larger period of time. You only need 10 bacteria to become infected, but for the most part, people who are infected aren't going to result in clinical symptoms. And it's going to take a while for these symptoms to develop because these bacteria are very slow growing. Most of the time, you're going to get infected and it's not going to result into an infection or clinical symptoms. You're going to try to phagocytose this with macrophages and it's going to become internalized. And that's going to cause a infection. And that infection can then either be resolved or it can go into further latency and recurrence where you're going to start to have these turbuncles form within your lungs. So this is where your macrophage has absorbed some of the, has phagocytosed some of the bacteria and it can't kill it and kind of puts it in a state of like panic. And we start to recruit other cells around it. We recruit other macrophages. We start to form fibroblasts in these like fibrous capsules around the infection. We're essentially like walling it off. And if it's still not cleared, we'll start to calcify that region and we get these calcified regions. And these are called turbuncles. And for the most part, this TB is really hard to clear and you need incredible regimens of antibiotics to actually clear it out. And it is found across the world. And so not everyone has access to those treatment resources, but it is incredibly fatal. And it spreads between people very easily. You can diagnose this where you inject a little bit of purified protein into the skin, and if it reacts, creates this swelled region, you'll be able to see if you are positive or negative. And the severity of it allows you to see how far along you are in that infection. You can see these turbuncles on X-rays, and then you can also use PCR to identify the bacteria. Again, we have really long treatments of antibiotics to really get through the infection. And with that, we are going to end this video. So I hope you had a great day. Bye-bye.